everyone. In the previous class, we discussed about methods of protein structure prediction. In that, we saw different experimental methods and computational methods. Whereas, in today's class, we will be discussing about X-ray crystallography. This is one of the experimental methods. First of all, we have to know what is X-ray crystallography is. X-ray crystallography is a technique where atomic and molecular structure of the crystal can be determined. When X-ray hits the crystal, it causes the diffracted beam to reflect in all the directions. And then more than 85% of the protein structure was determined with the help of the X-ray crystallography. Sir William Henry Burke was the first person to determine the crystal structure with the help of the X-ray crystallography. And then Watson Crick developed the DNA helix structure with the help of the X-ray crystallographic method in the year 1953. Now we have to know about the properties of X-ray. X-rays are electromagnetic radiations having a wavelength of 10 angstrom to 0.01 angstrom. And X-rays can penetrate to liquid, solid or gaseous molecule. In free space, X-rays can travel at the speed of 1,86,000 miles per second. It cannot be refracted or deflected or reflected by electric or magnetic field. They have the properties of interference, diffraction and refraction as like visible regions, and also they have germicidal or bactericidal effect, and also they are capable of producing an images on a photographic film. In an X-ray crystallography, why crystal structure is very important. The precise position of an atom or a molecule can only be determined if the crystal structure is in a pure and also in a well ordered manner. If the molecule does not have a very good crystal structure, then X-ray beam diffracted will be unpredictable, and from those data we will not get any appropriate results. And also if the crystal structure is in an ordered way, then the diffracted beam can give rise to a high resolution or high angle that can help us to find the 3D structure of the crystal. The principle of X-ray crystallography mainly depends on two main concepts. One is X-ray diffraction, another one is Bragg's law. We are going to see one by one. So X-ray diffraction. Diffraction is the slight bending of light when it passes through the edges of the object. And X-ray crystallography uses the uniformity of light diffraction to the crystal structure, and it helps us to determine the protein structure or crystalline structure. And also when X-ray hits the crystal, the electrons around the molecule will be diffracted in a different direction. This diffraction leads to some pattern formation. That pattern formation is called X-ray diffraction. Next concept is Bragg's law. Bragg's law was developed by Sir William Henry Bragg and Sir William Lawrence Bragg in the year 1913. It tells about the interference pattern of the different refracted beams. It helps us to find those interference pattern when X-ray is scattered when it hits the crystal. And it shows the relation of 2D sin theta equal to n lambda, whereas d is the spacing pattern between the diffracted beams, and theta is the incident angle, and n is the integer, whereas lambda is the wavelength of the atom generated during the process. Now we are going to see about the instrumentation part of the X-ray crystallography. The components in the X-ray crystallography are X-ray source, instant beam optics, receiver beam optics, goniometer, detector, and then finally sample holder. So X-ray source will be of monochromatic in nature, whereas in instant beam optics the condition will be stated or determined when the X-ray before hitting the crystal, whereas in receiver beam optics the condition will be monitored or determined after hitting the crystal. And then goniometer is the platform which holds the crystal structure, and also it holds the sample, optics and detectors. And then sample. So the sample for the X-ray crystallography should be in powdered nature, and also the size of the sample must be less than 10 millimeter. If the size of the sample is more than 10 millimeter, then crystal structure will not be appropriate. And then finally detector. So the number of X-rays scattered by this crystal can be determined or can be found with the help of the detector. Now we are going to see about the procedure for X-ray crystallography. If your sample of interest is protein, so initially you have to purify the protein and you have to check the purity of the protein. If your purity of the protein is greater than 99.99%, then you can go about the further processing. If your protein purity is less than 99.99%, then you have to again go for the another purification process. Once the protein is purified, then you have to precipitate the protein. So the precipitation of the protein can be done with the help of the suitable solvent. You have to use the suitable solvent to precipitate the protein. For example, in the case you can use 2-methyl 2,4-pentane-diol can be used as a solvent, is one kind of an example. Once you've done the precipitation, then you have to make the molecule to be supersaturated. So supersaturated is a condition where the concentration of the particular solution will be greater than the solubility rate of the particular solution in particular solvent, is called supersaturation. So supersaturation can be done by using the appropriate salt solutions. So you have to use appropriate salt to make your protein to be supersaturated. Then actual crystal growth. So crystal formation is of three main stages. One is supersaturation, nucleation, and then crystal growth. So supersaturation is the condition where the particular solvent or the solution have a greater solubility rate. And supersaturation can be done in many methods like evaporation, cooling, formation and addition. And nucleation is the formation of the nuclear formation over the crystal structure, is called nucleation. It's a very stable form. And then finally crystal growth. Crystal growth is the last stage of the crystal formation where nuclear growth will be stopped, whereas crystal will tend to form bigger in size, that is called crystal growth. In step 2, your crystalline protein is ready. Now you have to pass the X-ray to the crystalline protein. When you pass the X-ray to the crystalline protein, some X-rays will go along through the protein, some will deviate from your crystal formation. So you have to get to know all the diffracted beams, and also the crystal has to be in a rotated manner so that X-ray can be passed to all the edges of the crystal. The deviated beam will help us to understand the structure of the crystal, and also intensities and the position of the beam is also very important in identifying the crystal structure. In the step 3, the electron density map can be generated based on the diffracted beam patterns. And here the Fourier transform concept will be applied in order to get the electron density distribution of each atom or molecule in a crystal. And it plays a very important role. And also in this stage, resolution plays a crucial role in determining the protein structure or crystal structure. For example, resolution of the polypeptide chain will be 5 angstrom to 10 angstrom, whereas for the group of atoms will be 3 angstrom to 4 angstrom, and also for the single atom it will be 1 angstrom to 1.5 angstrom. Now we are going to see about the steps involved in X-ray crystallography. So in the first step, you have to purify the protein. The protein purity has to be greater than 99.99%. If the protein purity is below than that, then you have to again go for the purification process. And mostly hanging drop method is determined, used for the crystal formation. Once it is done, then X-ray will be passed to the crystalline protein. So once X-ray is passed to the crystalline protein, it will generate the diffracted beam. With the help of the diffracted beam, we will get the diffracted pattern. The diffracted pattern will go to the next stage, is called phasing. In phasing, we will get the electron density map. Here, Fourier transform concept will be generated or will be applied to get the electron density map. With help of that, we will get the 3D structure of the protein. Once your 3D structure of the protein is generated, then you can submit the model in the online databases. With the help of the online databases, many researchers can use those structure formation. Applications of X-ray crystallography. It is used to study the materials like metals, minerals, clay particles, semiconductors, as well as inorganic, organic and biological molecules. And also it used to find the electron density. So the atoms or the molecules present in the crystals can be determined with the help of the X-ray crystallography. And also their chemical bonding and their interactions and disorderness can also be determined. And also it is used to find the fine-grained minerals like clays, and mixed clays can also be found with the help of the X-ray crystallography. Finally, we can also find the structure and function of the biological molecules like drugs, proteins, some other chemicals, and DNA and RNA molecules. In the previous slide, we saw about the general application of X-ray crystallography. Now we are going to see about the application in the field of proteomics. So the first one is protein structure determination. So in protein structure determination, we are able to find the all possible amino acids present in the particular protein can be studied. And then protein interaction, it means the interaction of two or more proteins. So the main and major amino acids present in the interaction can be studied in the protein interaction. And then conformational studies. So in the conformational studies, we can get to know the atoms involved in the protein formation or molecules involved in the crystal formation can also be studied, and also we can get to know the chemical bonding interactions too. And finally, enzyme catalyst determination. So in enzyme catalyst determination, we are able to find the active sites present in the enzyme and also the major amino acids present in the active sites, and then catalytic efficiency, catalytic ability, and also catalytic interaction of the enzyme can be studied in an appropriate manner by using the X-ray crystallography. So finally, we are going to see about the advantages and disadvantages of the X-ray crystallography. So what are the advantages of the X-ray crystallography? This method is very cheap and simple, and we can able to predict the 3D structure of the crystal or protein with help of the 2D structure. And then it has a very good atomic resolution, and it can be used to determine the large structure of the protein or any other material. The disadvantage of this method is the sample has to be in a crystalline form. If the sample is not in a crystalline form, then we cannot use this method. And also, if it is not in a crystalline form, we will not get the proper diffraction pattern. And then this method is not useful for all biological molecules, especially it is not useful for the membrane bound proteins. And also the type of material or protein used in the X-ray crystallography is very limited when compared to the other methods. These are some of the disadvantages in this method. We came to the end of the session. I hope you understand about X-ray crystallography. In the next session, we will be discussing about NMR spectroscopy. Thank you all.